Why is transfection a crucial technique in biomedical research? This might be a question that pops up in your mind quite often. Well, let's dive into the answer. Transfection, in the simplest of terms, is a method used in laboratories to introduce foreign genetic material like DNA, RNA, or proteins into cells. This technique is a cornerstone in various scientific and medical fields. It's like the Swiss Army knife of molecular biology, versatile and essential. One of the key applications of transfection is in gene function studies. By introducing specific genetic material into cells, researchers can observe the changes in cellular behavior, providing valuable insights into gene functions. It's like giving a new script to an actor and seeing how they perform. Transfection is also instrumental in protein expression. By introducing the gene encoding a particular protein into cells, researchers can produce that protein on demand. This is incredibly useful in therapeutic protein production, where large quantities of a specific protein are required. Moving on to gene therapy, transfection plays a vital role here as well. It's used to deliver therapeutic genes into patient cells to treat genetic disorders or diseases. It's akin to sending in a specialist team to fix a problem. Functional genomics is another area where transfection shines. In these studies, researchers aim to understand the role of specific genes in cellular processes. Transfection is the key to unlocking these genetic secrets. Lastly, in drug development, transfection is used to screen the effects of potential drugs on specific cellular pathways by manipulating gene expression. It's like a rehearsal before the main performance, ensuring that the drug works as intended. So as you can see, transfection has wide-ranging applications in scientific and medical fields, making it a crucial technique in biomedical research. It's a tool that, when used correctly, can lead to breakthroughs that revolutionize our understanding of biology and medicine. Now that we understand the importance of transfection, let's delve into the various methods used to carry out this process. Let's start with viral transfection, a highly efficient method of introducing foreign genetic material into cells. Viral transfection involves using viruses as vehicles or vectors to deliver genetic material into cells. Now this might sound a bit alarming, but don't worry, these viruses are engineered to be safe and won't cause disease. Firstly, let's talk about adenoviruses. These little guys are pretty versatile, being efficient for a wide range of cell types. They work by entering a cell and delivering their genetic cargo without integrating into the host genome. This characteristic makes them perfect for transient expression, that is, short-term expression of the introduced genes. However, the downside is that they can't be used for long-term studies because the introduced genes won't be passed on when the cell divides. Next up, we have lentiviruses. These viruses are a bit more specialized. They can infect both dividing and non-dividing cells, making them incredibly useful in a wide variety of research settings. Unlike adenoviruses, lentiviruses integrate their genetic material into the host genome, which allows for stable, long-term gene expression. This means the introduced genes can be passed on to daughter cells when the host cell divides. However, this advantage can also be a disadvantage, as there's a risk of disrupting important host genes when the viral genes integrate. Both adenoviruses and lentiviruses are highly efficient at delivering genes into cells, but they each have their pros and cons. Adenoviruses are great for a wide range of cell types and allow for transient gene expression, but they can't be used for long-term studies. Lentiviruses, on the other hand, can infect both dividing and non-dividing cells and allow for stable, long-term gene expression, but there's a risk of disrupting host genes. Viral transfection methods, while highly efficient, may not always be suitable. This brings us to non-viral methods of transfection. But before we dive into that, let's take a moment to appreciate the beauty of science. Always evolving, always giving us new tools to understand the world around us. Stay tuned. Chemical methods of transfection are often more versatile and less complex than viral methods. Delving into the realm of chemical transfection, we have three prominent methods: calcium phosphate transfection, liposome-mediated transfection, and polymer-mediated transfection. Each of these methods leverages the power of chemistry to introduce genetic material into cells, but they do so in unique ways. Starting with calcium phosphate transfection, this process involves creating a precipitate of calcium phosphate and DNA. The cells then engulf this precipitate, granting the DNA access to the inside. This method is relatively simple and cost-effective, making it a popular choice for many researchers. However, it's not all rosy. The downside is that it's a bit unpredictable and can be toxic to some sensitive cell types. Next, we have liposome-mediated transfection, also known as lipofection. Here, the DNA is enclosed within a lipid vesicle, essentially a tiny bubble, which then merges with the cell membrane, delivering the DNA into the cell. Liposomes are biocompatible and can be easily modified, offering a high degree of control and flexibility. Yet, they can sometimes be inefficient and may require optimization for different cell types. Finally, there's polymer-mediated transfection. This method uses positively charged polymers to form complexes with the negatively charged DNA. These complexes are then taken up by the cells. The advantage of this method is that it can be very efficient, particularly for hard-to-transfect cells. However, the downside is that the polymers can be cytotoxic, and the transfection efficiency can vary. In summary, chemical methods of transfection provide a range of options with varying degrees of complexity, efficiency, and cytotoxicity. They offer a valuable toolkit for researchers seeking to introduce foreign genetic material into cells. While chemical methods are versatile, there are other non-viral methods that employ physical and mechanical principles. Scene script. Physical methods of transfection utilize mechanical forces or electrical fields to introduce foreign genetic material into cells. First up, we have electroporation. This technique applies a brief electrical pulse to cells, creating temporary pores in the cell membrane through which DNA can enter. Electroporation is efficient and can be used on a variety of cell types, but it can also be quite harsh, potentially leading to cell death. Next, let's talk about microinjection. As the name suggests, this method involves using a fine micropipette to directly inject DNA into a cell. Microinjection allows for precise control, as you're literally putting the DNA exactly where you want it. However, it's also quite labor-intensive and not suitable for large-scale experiments. Third on our list is biolistic transfection, also known as gene gun method. This technique uses high velocity to shoot DNA-coated particles into cells. Biolistic transfection can be used for a wide range of cell types, including plant cells, and it doesn't require cells to be dividing. On the flip side, it can cause significant damage to the cells and is not efficient for large numbers of cells. Finally, we have nanoparticle-mediated transfection. Nanoparticles can encapsulate DNA and deliver it into cells. This method is particularly promising for targeted gene delivery in gene therapy applications. However, the technology is still developing, and the potential long-term effects of nanoparticles are not fully understood. Each of these physical methods of transfection has its own strengths and weaknesses, and the best choice often depends on the specifics of your experiment. Are you working with a hard-to-transfect cell type? Do you need to introduce DNA into a large number of cells? Do you require temporary or stable gene expression? These are the kinds of questions that can guide your choice. The choice of transfection method depends on various factors, which we will discuss next. Choosing the right transfection method is a crucial step in any experiment or therapeutic application. When selecting a transfection method, several factors come into play. First and foremost, the cell type. Some cells, like primary cells and stem cells, can be challenging to transfect, requiring more efficient methods like viral transfection. On the other hand, easily transfected cells, such as HEC293 or HeLa cells, can be manipulated using less invasive non-viral methods. The desired duration of gene expression is another key consideration. For transient expression, where the gene of interest is expressed for a short period, adenoviral vectors or chemical methods like liposome-mediated transfection can be ideal. For stable long-term expression, lentiviral vectors, which integrate into the host genome, may be the method of choice. The nature of the genetic material to be introduced is equally important. For example, if you're working with plasmid DNA, chemical or physical methods could suffice. However, for more complex genetic materials such as long RNA sequences or entire genes, viral methods might be more effective. Let's paint a clearer picture with a few examples. Suppose you're studying gene function in a hard-to-transfect cell type like neurons. Here, a lentiviral vector could be your best bet. If you're interested in producing a protein in large quantities for therapeutic purposes, you might opt for liposome-mediated transfection in a cell line like HEC293, known for high protein yield. Or perhaps you're exploring gene therapy approaches for a genetic disorder. In this case, nanoparticle-mediated transfection could offer the targeted delivery you need, minimizing off-target effects and maximizing therapeutic efficacy. Every experiment is unique with its own set of requirements and constraints. The key is to understand the strengths and limitations of each transfection method and align them with your specific needs. Understanding the importance of transfection and the various methods of carrying it out equips us with the knowledge to make informed decisions in our research endeavors.